Hello there, and thank you to the good people at JB Moneybags Incorporated for listening to our grant proposal this evening. We're very, very excited to be discussing the inhibitory effect of green glow funkadelicis's green fluorescent protein on the BRAF mutation V600E in malignant melanoma xenografts. My team consists of myself, Michael Fralick, Julika Job, Madison Jochum, and Joaquin Sandoval. First, we will be discussing melanoma in the United States, why it's important, why new treatments needed. We will then be discussing the green fluorescent protein, where it comes from, and why does it have potential in treating skin wounds and skin cancer. Then we'll be discussing a little bit about the preliminary experiments used to isolate and molecularly clone GFP. Then we'll be discussing the AIM proposal we have. The objective of it, its justification, the design of our experimentation, the expected outcomes, and some alternative approaches. First, I will be talking about melanoma. Skin cancer is the most prevalent form of cancer in the United States. 9,500 daily new cases of skin cancer are diagnosed. 1% of these cases are melanoma. Seems like a small amount. However, when you consider that melanoma is the most deadly of the skin cancers, doesn't seem so small. Over 100,000 yearly melanoma cases occur in the United States and around 7,000 deaths at a 7% fatality rate. There is no consistent or reliable treatment. There exists some chemotherapy, immunotherapy, and cutting out the skin cancer. However, these therapies do have side effects such as inflammation and nausea. They aren't proven to work every single time. However, some types of treatments, such as focuses on proteins and genes, have tended to work with a higher success rate. However, these types of inhibitors of proteins and genes do have some resistance that has developed against their treatment properties. Change must come. And we believe this change can come from the green fluorescent protein. Now, we are very excited to discuss this because it comes from a plant in the Amazon jungle of Brazil. It is called green glow funkadelicis. And this plant has been used by many indigenous tribes for many generations to treat skin wounds. It's actually been combined with cannabidiol or CBD to use in treating skin wounds. Now, our brains are working, maybe this can be used to treat skin cancer. That is what we will be discussing. However, there's limited harvestability of GFP. It requires cloning. Thus, we will be using our experimentation to isolate and clone the green fluorescent protein because the future of melanoma treatment lies within. Now, first, we're going to be discussing the preliminary experimentation of this. We will collect the green funkadelicis plant sample. We will extract and isolate its DNA using designed primers. We will linearize a plasmid so that we can create a recombinant plasmid after ligation of digestion products and our linear plasmids. Now, the reason we want to create these is so that we can transform these recombinant plasmids into E. coli cells to be used for further experimentation. Now, first we must determine whether a transformation can successfully occur under selective mediums, such as luria broth, ampicillin, and arabinose. If successfully transformed, E. coli colonies will develop on agar plates. And as we see right here, we want these colonies to be successfully transformed so that our recombinant plasmids stay around. They stick around. Our P-glow and P-bad, we want those to be around. And as we can see right here, that is what it will look like if they remain. This is the GFP, the green fluorescent protein. It is fluorescing. Next, we will isolate the DNA and amplify the DNA using PCR. We will confirm the presence of P-glow using gel electrophoresis and PCR. Then we will sequence the DNA, as we can see right here, this nice flow chart, and then we will inoculate the confirmed E. coli colonies into liquid selective mediums, and then we will purify them using hydrophobic interaction chromatography. Now, all of this is required so that we can perform our main experimentation, which is our AIM proposal, which we will be discussing. I'm going to pass it along to Joaquin, and he'll be talking to you about that.
Okay, well, thank you for addressing melanoma, GFP, and our preliminary experiment, Michael. We're now going to shift gears and focus on our aim for this funkified grant proposal, which will involve investigating the synergistic effects of GFP and CBD on the expression levels of phosphorylated MEK protein in musculus. Let's get into it. So our protein of interest is MEK, and it is involved in the MAPK, or the mitogen activated protein kinase pathway. So this pathway essentially allows an extracellular signal to activate a cascade of proteins that activate certain transcription factors that yield cellular response. So the activation in this pathway happens to be phosphorylation. In short, a ligand binds to the extracellular receptor, which activates RAF. RAF then phosphorylates and activates MEK, okay, which does the same to ERK, which does the same to a transcription factor, which induces a response such as proliferation, development, etc. Okay, so right away, you might be able to see that dysregulation of this pathway could lead to cancer. So for example, a mutation commonly found in melanomas, in fact, in nearly half of melanomas, BRAF V600E is an additional component to this pathway that overactively phosphorylates MEK and contributes to an overactive MAPK pathway. Okay. So our objective is to inhibit the BRAF V600E product, thereby preventing the phosphorylation and overactivation of MEK protein. So this would lead to a lower degree of phosphorylation of MEK, yielding lower phosphorylated MEK levels, and disrupt an overactive MAPK pathway, contributing to progression of melanoma. Now, there are BRAF V600E inhibitors that are being utilized right now, and I'll talk about their side effects in a bit, but we hope that our drug coming from a natural source, such as will yield less abrasive side effects. Okay, so we know that abnormal signaling of the MAPK pathway has been identified and is linked to uncontrollable tumor growth. So the BRAF V600E mutation has also been linked directly to increased cell proliferation in melanoma, and it also increases the risk of recurrence. So there have also been several instances of melanomas containing this mutation that show resistance to MEK and ERK inhibitors. So we believe that targeting the root of this problem, the mutated BRAF protein, directly will be the most effective method for disrupting this hyperactive pathway. All right. So there are several mutated BRAF inhibitors such as vemurafenib and dabrafenib that are available and are currently being used to treat melanoma patients. The only problem with these medications is that they are accompanied by many unwanted side effects. Side effects of vemurafenib include blistering, peeling, or loosening of the skin, bloating or swelling of the face, bloody urine, blurred vision, etc. Side effects of dabrafenib include reddening of the skin, hair loss, fever, chills, joint pain, etc. So in short, these BRAF inhibitors may disrupt the progression of melanoma, but do so in a harmful manner to the patient. So we are confident that our version of the BRAF inhibitor will not be accompanied by such detriment. So inspired by the medicinal methods of the indigenous of the Amazon, we decided that we would like to combine our GFP drug with CBD. So CBD is known to have anti-tumor effects, and several studies have suggested that CBD may induce cell death and inhibition of growth in cancer cells. Additionally, it has been shown that CBD has anti-inflammatory properties, and they are helpful in treatment in cancers that produce inflammation, such as in melanoma. Additionally, they also promote the uptake of cytotoxic drugs into malignant cells. So GFP and CBD is the way to be, and I'm going to pass the mic to Julika. Thank you.
Thank you so much, Keen. So here you can see a brief overview of our AIM-1 research design. We're going to start out by creating a xenograft with human malignant melanoma cells that contain the BRAF V600E mutation. We will then let the tumor tissue grow over the course of 6 weeks before performing a biopsy. This biopsy will allow for the tumor tissue to be analyzed using DNA sequencing. After this step, we're going to start with administering our treatments. The phosphorylated MEK levels are going to be checked on a regular basis using a Western blot. And this Western blot is also going to be used to check the expression levels of C-reactive protein, which is an indicator for inflammation. Regarding the xenograft, we decided to use a total of 15 mice. We decided on M. musculus simply because they have already previously been successfully used in BRAF inhibitor studies. Therefore, we thought it would be appropriate for this particular study to use them as well, especially because we are going to compare it to a already FDA-approved BRAF inhibitor. For this particular experiment, we are going to transplant human malignant melanoma cells under the skin of those 15 mice, and we will then let those develop over the course of 6 weeks into those cutaneous malignant melanomas. After 6 weeks, we are going to perform a biopsy. We will collect the sample and isolate the DNA. We will then perform PCR before the DNA gets sequenced. This step is really important because here we have to really ensure that the BRAF V600E mutation is present in all tumors before we start the treatment. Regarding the treatment, we did decide to administer weekly treatments of 6 milligrams of GFP and 2.5 milligrams of CBD per kilogram of mouse body weight. Here you can see our 7 different groups of mice. 5 of those groups have the xenograft, and 2 of those groups consist of healthy mice. Each group consists of 3 mice. So here, the very first group with the xenograft is going to receive GFP and CBD together. The second group is going to receive GFP, and the third group CBD. It's important that we give those to the mice to see the independent effect of each component to really be able to trace those back to its origin, basically. And the same thing is for the side effects. It's really important that we see the impact each component has on the mice individually. The fourth group is going to receive a already FDA-approved BRAF inhibitor. This BRAF inhibitor has to be administered at 10 milligrams per kilogram of mouse body weight due to its relatively low bioavailability. And the fifth group is going to receive simply a saline solution. Amongst the healthy mice, we decided to administer one group with GFP and CBD to see what are the impacts of GFP and CBD in combination on a healthy individual. And then the other group is going to receive the saline vehicle because we want to basically monitor what are the impacts of the laboratory conditions overall on the mice. To monitor the phospho-MEK levels and the C-reactive protein levels, as previously mentioned, we are going to use a Western blot. This is going to be performed 5 times. The first time right before the first round of treatments, then the second time on day 14, the third time on day 30, and then the fourth time after 60 days, and the last time after 120 days. For the Western blot, we have the standard procedure. We have to isolate the proteins. We will perform SDS-PAGE to separate them based on their molecular properties. And then we will transfer the proteins onto a nitrocellulose membrane. And then we are going to perform the immunoblot. Here we have listed the specific primary and also secondary antibody that we chose to measure the phospho-MEK expression levels. And then over here, you can see the specific primary and secondary antibody that we chose for the C-reactive protein. And this will allow us to really compare it over the total of 4 months, like to see how those expression levels change. For both immunoblots, we will use chemiluminescent detection, and Maddie is going to go over our expected results in the next part.
Thank you, Julika. So on day 0, prior to any drug injections, samples will be taken from the tumors of each group of mice, and a Western blot will be conducted to measure the expression levels of both phosphorylated MEK protein as well as C-reactive protein, otherwise known as CRP. Samples are then taken periodically throughout the experiment, and Western blots will be conducted. The lane closest to the left, labeled MWN, stands for our molecular weight marker. In this case, we'll be using a pre-stained protein ladder. The lane labeled vemurafenib contains samples of mice treated with one of the control variables, the inhibitor vemurafenib. This will give us an idea of how well the GFP holds up in comparison to a BRAF inhibitor already currently on the market. The next lane, labeled saline vehicle, contains samples of mice treated with strictly saline, serving as our other control variable. The next 3 lanes contain samples of mice treated with experimental drugs containing GFP and CBD combined, GFP alone, and CBD alone. The phospho-MEK expression level visualization would result in markings at around 43 kilodaltons, so right around here. And the CRP expression level visualization would result in markings at around 120 kilodaltons, so right around there. This slide is representative of the best possible outcome from this experimentation. It's our expectation that the GFP drug eventually surpasses the BRAF V600E inhibition levels of the leading FDA-approved inhibitors, such as vemurafenib, as they have been known to eventually occur resistance in melanoma patients. This would result in visualization of the phospho-MEK levels being lower or non-existent in our GFP drug lanes than in our control. This outcome would lead to more testing and eventually human trials in order to get this improved BRAF inhibitor on the market. So as you can see here on day 0, phosphorylated MEK levels are about equal in all lanes. But as day 14 rolls around, both the GFP drug and the vemurafenib have inhibited BRAF, leading to lower phospho-MEK levels. On day 120, however, we expect to see some resistance to vemurafenib, resulting in a bit higher phospho-MEK expression. Yet we expect significantly lower phospho-MEK levels in the groups that received GFP and CBD together and GFP alone, since we do not expect the melanoma cells to develop resistance to the inhibitory effect of GFP. This next figure represents a second possible, but less likely outcome, in which GFP did regulate expression levels of phospho-MEK compared to the saline vehicle. However, in the 4-month timeframe, it did not decrease expression levels as much as vemurafenib, the leading FDA-approved inhibitor. This would demonstrate that while the GFP drug works slightly better than a control, it would not make sense to continue this route of testing. It would be suggested that new molecular pathways in regard to GFP may want to be explored. The last and least expected outcome for our phosphorylated MEK Western blot would be if expression levels were heightened upon recurrent administration of the drug. There's no evidence leading us to believe that this would happen. However, if the GFP drug results in higher phospho-MEK expression than its saline vehicle, a halt of the trial would be recommended, and inquiry into the potentially cancerous effects of GFP would be recommended. To conduct analysis on the combined effects of GFP with CBD, a Western blot will measure the amount of CRP before injections and after 120 days of both CBD combined with GFP and CBD alone. Due to the outstanding amount of evidence on the anti-inflammatory effects of CBD, as well as its combined usage with GFP by the Amazonians, it is safe to assume that the most likely outcome is a decrease in CRP expression after 4 months' time. Anything else would be extremely unexpected. The last slide I'll be showing you today is the expected results of the CRP expression levels of 2 control groups of healthy mice. One group of healthy mice will be given the saline vehicle to ensure that there are no other unseen factors affecting the trial, and one group will be administered the combined GFP and CBD drug. This ensures that there are no adverse effects to the drug, and it's our expectation that the healthy controls will show an insignificant change in CRP levels before and after the trial. Next, Joaquin will be discussing potential problems and alternative approaches within our AIM-1.
So as far as our aim goes, we have identified some potential problems, but we have alternative approaches to take if we were to ram into them. One problem would be an increase in phosphorylated MEK levels. So if we were to see this, we would simply increase the GFP concentration used from 6 to 10 milligrams per kilogram of mouse body weight. We would take the same approach if we were to see inflammation increase by increasing the CBD concentration to 5 milligrams per kilogram of mouse body weight. Now, the last thing is that the BRAF V600E mutation might not be representative enough to enable this drug to be used for all melanoma patients, basically because this mutation occurs in really only half of melanomas. But we are still hopeful that our GFP drug treatment can remain as a secondary measure and can be applied to other proteins or genes that are adversely affected by malignant melanomas. So in conclusion, we think that our natural drug may be more effective than current treatment methods out there and may yield less harmful side effects. We will also ensure that JB Moneybags Incorporated will be accredited with the proper funding acknowledgement statement. Thank you so much. Here are our references, and we thank you for your time and consideration.